Hi, everyone. Arslan Arif here with Endpoints News, and thanks for joining us for Integrated Clinical Development and Supply, breaking down the early development journey of a small molecule. We're sponsored by Catalent, and I'm excited to introduce today's panel. Joining us, we have Stephen Tyndall, the Director of Science and Technology at Catalent, and Mark Prutt, Head of Early Drug Development at NGT Biopharma Consultants. If you have any questions during today's discussion, just hit the Q&A button at the bottom of your screen. We've got some time at the end of the webinar to answer your questions. This is going to be available on demand tomorrow to view or to share it with your colleagues. And now I'm going to pass it over to Mark to get us started. Mark?
Thank you, Arslan. So I'm going to take a few moments and talk about how and when to think about integrated drug development, specifically for small molecules. As a disclaimer, I am an independent consultant. I also work for NGT Biopharma Consultants, but the views that I'm presenting today are exclusively my own. I will set the stage with a high-level scope of drug and CMC development. I will talk about what non-integrated drug development looks like. I'll give you an example of that. I will then focus on integrated drug development, also with an example. I will discuss briefly the trade-offs between these two development approaches, and I will leave you with a few key takeaways. So first, at its very basic, drug development is really about answering two simple questions. Is the molecule safe? And is it efficacious? That is, does it work? And every drug company wants to answer these questions as quick as possible with the minimum of investment in either resources or money. At the same time, within that same company, there's a CMC development group that is going to be answering a different set of questions. The first is, how can we take that lead molecule? How can we scale it up? How can we make it into a formulation that's suitable for a first-in-human trial? And how can we get that done as quickly as possible? That's the short-term approach. Longer-term view is, can we make that product and manufacture it at scale? Can we do it safely, reproducibly? Can we make it with consistent quality and at an acceptable cost? And a CMC organization is going to try and do this while staying off the critical path. Now, in practice, in a non-integrated sense, a typical biotech approach, if you will, is to look at these as individual activities. So they're sequenced in a way that they get done in advance of a key milestone. And here I've outlined a filing of an IND and the start of a phase 1 study. So with a biotech company does not have these resources in-house, so they will work with a variety of different contract manufacturing development organizations across the world, typically, to complete each of these individual activities. And as long as they're done on time and one is handing off to the other, a milestone and the appropriate timing can be met. So for example, process chemistry group will develop a synthesis for scale-up and will make the API. A portion of that API will be delivered to a CRO who will conduct the preclinical tox studies. The remainder of that API will be transferred to a formulation development group where they will look to develop a first-in-human formulation. They will scale up those clinical supplies. Those clinical supplies will then be delivered to a packaging and distribution group who will package that and then distribute it across the world and into the appropriate clinical sites. Behind all this, there'll be analytical development work ongoing, probably supporting 3 of these individually. So there'll be an analytical group embedded with the process chemist. There'll be another analytical group supporting the formulation development, and probably a third one supporting package development. Could also be the case that there's a separate analytical group altogether that is responsible for final clinical testing and releasing of the drug. So in a non-integrated sense, you just do each of these individual activities in time, and you get to your milestone, in this case, a first-in-human study. Then you just repeat those activities. You just do it again. You make more API, formulate, scale up again, package, distribute, analytically test. You do that, you can get to a phase 2 study. You do it a couple more times, you'll get through phase 3, you'll file your new drug application, and you'll be able to get approval for your drug. However, the reality is that repeat process simply doesn't work. It's inevitable that something will come up that prevents just the simple repeat of those activities. So consider, for example, API synthesis. So the process group is going to make typically a non-GMP gram scale quantity drug to support preclinical tox studies. They're going to do this. Then they're going to turn around and scale that up under GMP conditions and make on the order of a couple kilos of API that's going to be converted into clinical material. It's inevitable that there will be changes in that process used to make that API as you increase the scale. So let's say, for example, that alters the impurity profile. Now, if the API group just simply hands that on downstream to the tox folks, or to the, in this case, to the formulators, without a clear understanding of those changes and the impact it has on the impurity profile, that can set off a cascade of activities. So if it's just simply now we have new impurities or different impurities, we might have to develop new analytical methods to test for those. If we have to control the impurities, that is, if we have to bring them down to the levels that they were qualified in the preclinical studies, the tox studies, then work is going to go back to the process chemist to either improve the process and or purify the material that's already been made. In a worst case scenario, those new impurities, or the higher levels of impurities, as it were, are going to stay with us now, given the fact that we've changed the synthesis. Then we're going to have to go back and requalify those. So just the simple case of an API group doing a scale up can have a cascade effect across other functional areas, analytical, back to the process chemistry, even the tox group, to do additional work that really wasn't planned for, because there was not a good link between the organizations and the understanding of what's going on and the changes in a process development. And there's a multitude of CMC-related changes that can occur during development, and some of these will. And I'm going to highlight a couple here with the possible consequences of or impacts to the other functional areas. So for example, going in development, if your compound is capable of forming a salt, you may be bringing an initial salt in, but you may find for various physical chemical property improvements, it's worthwhile to switch to a new salt. If you do that, that's a new molecular entity in the eyes of the health authorities. So you're going to start over. Polymorph. So changes in polymorph or crystal form can occur. If they occur during development, that's going to have an impact on stability work. Maybe the case that you're going to have to develop process controls to be able to control and give the desired polymorph that you want. Probably going to have to go back and tox qualify a new polymorph. New analytical methods are needed. It's going to have an impact on bioavailability, which in turn could affect your dose selection, and that's going to impact the formulators and how they're going to use that drug. Particle size distribution. Quite common that the particle size can change depending on the scale. Maybe the case that you need to control particle size, again, has impacts on bioavailability, process controls, and on the formulation group. So all of these things, there'll be new analytical methods that are going to be developed along the way. That has consequences. Food effects. If you get into a human study and you find out there's food effects, you're going to have to go back and adjust the formulation to account for that. The formulation itself will change during development. What goes into a first-in-human study will not be the final formulation that is ultimately released and approved for market use. And then primary container has an impact on stability and shelf life. So the point is, the repeat process doesn't really work all that well, because there will be changes along the way that has to be taken into account and managed accordingly. So really, in an integrated sense, in integrated drug development, it considers all these activities holistically and how they link to each other. So API is directly tied to formulation, what gets done there. Formulation should be interacting closely with packaging to make sure that the formulation is appropriately supported by the right package. And analytical should really be one analytical group underpinning all of this, similar methods, similar understanding. And in the end, this directly links to the patient. But the idea is to try and build deep knowledge within not only each of these functional areas, but across the entire CMC landscape. And my contention is this is going to lead to better decision-making along the way and ultimately a better product for the patient. So now reconsider that same example I just talked about. Instead of API working in isolation, it's now closely communicating, collaborating with analytical to understand process changes and the impact on that and what that does to that impurity profile. And the API group is also working closely with the formulators. Is the physical form changing? Are we seeing changes in polymorph? Are we seeing changes in particle size? How does that benefit or detract from the formulation that's being developed? Formulation does not operate in isolation, in a vacuum. All of the work that they do should be measured by in vivo performance. So that's supported by PK studies. As I said earlier, the formulation also needs to work with the packaging. Is it going to be, what is the packaging that we want for clinical trials? Is it bottles? Is it blisters? Is the drug hydroscopic? And we've got a hydroscopic formulation, so we need to think about putting in desiccant in the bottles or desiccant in the blisters. Those need to work in concert. And analytical, as I said, should be tied to very closely to each of these functions and supporting them together. However, it seems like a lot. It seems like a lot of planning, a lot of thought goes into this. Yes, it does. But with some advanced planning and a little bit of resource investment, an integrated approach can have little or no impact on critical path. So you can get it done without worrying about upsetting your overall timelines. There are trade-offs, however. In a non-integrated sense, in a way, it plans for failure. It's a fail-fast approach, and that's not necessarily a bad thing. There are times where, quite honestly, you want to get a quick kill in, you want to get some answers, and so a non-integrated approach will get you there faster. Integrated approach takes a little bit longer view. It tends to plan for success, assuming that the molecule is going to continue to go through each of those key milestones, see a positive result, and then move forward to ultimately an NDA. It also allows you the ability to fail smartly, and I'll come back to that in a couple of slides. A non-integrated approach is fast to the first milestone, but it will be slower overall for the reasons I just cited. Additional work, things will come up, you have to go back and investigate further. In an integrated fashion, it's going to be lower cost and resources overall, but because you're doing some advanced work and probably a little bit more work to build that deep understanding, there is the risk that that work could be lost or not needed if you see early attrition with the product. I suggested that non-integrated is short-term horizon. It really is just looking to the next milestone. More importantly, integrated takes a longer-term view, looks at the full cross-functional holistic product view and builds better knowledge and understanding of the product itself. And then obviously in an integrated way, it really does rely on very effective cross-communication and collaboration between the functional groups. The process chemists have to be talking to the formulators. The analytical folks have to be talking to all of the functions. Now, fail smartly. I mentioned this briefly. There was a paper last year that talked about the failure to fail smartly, and it was looking at the preventable causes of late-stage failures. And they looked at three factors, or considered three factors: an expertise-based failure, execution-based failure, and knowledge-based failure. And underneath here is a couple of key points out of that. And you can see in yellow, I've highlighted some that I believe can be addressed by taking a more integrated approach to drug development. So they cited inadequate drug development know-how. I think if you link the functions together, you can solve that. There's been problems with scale-up. Again, communication, inadequate communication. That was highlighted. I've mentioned that. Problems with data interpretation, or simply just poor decision-making. And I think by better linking the functions together, you're able to avoid some of these preventable causes of product failure. So a couple of key takeaways. I'd like to make it clear there isn't an either-or option. It isn't one or the other. In fact, you can do a combination of those. Some cases, as I suggested, a non-integrated approach might be the right thing to do versus a longer-term integrated view. Really, the answer is in tailoring the development approach, not only to your molecule, but also your business needs. More importantly, though, it's this idea of using data-driven decision-making, so building strong, deep knowledge of your product, and use that information for product progression, or in this case, failing smartly, use that for product termination. And with that, I'm going to turn it over to Stephen.
Okay, thank you, Mark. And it's a great pleasure for me to talk today about Catalent's approach to integrated development for oral small molecules. Catalent is well known as a CDMO with development and manufacturing expertise, but few people are aware that our scientific advisory service that helps prevent some of the pitfalls that Mark has described. I'm going to show you what is scientific advisory, how Catalent can help you with three key issues: 1, improving communication; 2, improving outcomes; and 3, shorten timelines with an integrated approach. GMP requirements are important and help ensure the safety and efficacy of medicines that a patient is taking. And of course, regulatory requirements help drive the overall project through important milestones like clinical studies. But in small companies, project timelines tend to be set with a combination of assumptions and heroic optimism, like the regulatory process drives every project irrespective of the API. That means clinical project teams must find and mitigate any issues along the way, often from minimal data and without breaking the bank or extending critical dates. This makes it vital to work with teams with deep expertise who've been through the process many times with more challenging molecules perhaps, and who are expert in recognizing issues hidden in limited data sets in order to redirect the proper studies. If scientific issues begin to accumulate and the risk-benefit balance starts to change, companies need to communicate and respond quickly, which means it is critical to be able to recognize issues early, especially for companies that only have one molecule in their pipeline. The major problem is that more and more molecules have issues which themselves aren't fully manifest until after the phase 1 study and possibly beyond, due to the way that development work is minimized these days. This means it's important to employ best scientific practice throughout the development and not just rely on pair-of-hand CDMOs to make and supply in response to an RFP. In this way, it becomes possible to identify or predict critical issues that might be hiding in the limited data and to flag them as soon as they become apparent. To emphasize this pictorially, in the right picture, late phase, we have experience, understanding, validated or qualified methods and processes that altogether help us determine whether we can reproducibly manufacture a drug product within GMP and to recognize trends and issues. That does not guarantee clinical success. In the left picture, early development, we need to balance the needs of GMP with the data collection process and where methods are prone to higher variability. If we employ best scientific practices, we can reduce variability whilst at the same time providing better interpretation of data in order to make robust conclusions in the absence of expertise on that molecule in hand. Working with a large CDMO like Catalent can be a good option here, bringing focus onto tactics that work from handling hundreds of other molecules and from unpublished studies. The key transition point for many people is usually after phase 1, what venture capitalists refer to as proof of concept, but it could be whenever dosage form design is needed. When engaging with a customer who would like help, a Catalent scientific advisor is allocated to perform a broad review of information, then dig more deeply into perceived or real issues, provide advice, and provide a summary recommendation. If Catalent is selected as the provider, they transfer information to the product development team for execution. If needed, the advisor can stay with the project to oversee the transition from drug substance to drug product, or if there are any surprise findings. Many people understand how indication, for example, can inform the broad needs of a project, and it's certainly useful to know who the intended patient population might be in order to rule out or rule in some approaches. But it's best scientific practice to also understand questions like, what is the mechanism of action? And where is the target? As these can unlock other issues that need to be addressed. For example, if the target is a receptor in the brain, the drug will need to cross the blood-brain barrier. Or if the target is parasite in the GI tract, it may not be beneficial to have much, if any, systemic circulation. Also, in some cases, the first indication may be selected as a foundation for future expansion into other therapeutic areas in the future. In order to be a better partner, these considerations are important and best to discuss in the very first meeting, even when there is a sense of urgency to get going quickly. Once all this information is available, the scientific advisor can give advice on the best team to work with and oversee the API to drug product transition in a patient-friendly dosage form. In early development, this typically starts by reviewing existing data at whatever stage and recommending the scope of work to support the technology selection or even to challenge the planned route of administration. In early development, Catalent Scientific Advisory is available to perform a kind of triage in order to provide a recommendation about the best technology or to screen multiple options in parallel. This is done with an agnostic approach, selecting from the broadest range of technologies. if the data set is incomplete, suggesting options for next best action. The overall goal is to help identify challenges and opportunities for each drug candidate and introducing Catalent's product development team to explain the advantages and disadvantages of each drug delivery technology. Inside Catalent, the global scientific advisors meet together to stay abreast of latest developments, discuss emerging issues, and troubleshoot problems together, as well as engaging with product development to distill our combined expertise into our protocols and our evaluations, bringing access to our global network to bear on any problem. After deciding on the molecule, which in itself may not be such a clear choice, its structure and polymorphic form drives its properties, and the differences can be huge. We talk about know your molecule, where our evaluation is revealing what was always going to be the case once the form was selected. By understanding a molecule, we can usually mitigate any liabilities with careful formulation and provide advice on what would be the cost and how long it would take. But it's also important to monitor properties during API development, as Mark previously mentioned, as things often change. For example, improvement to purity that typically happens after GLP tox and before phase 2 can have a serious negative effect on solubility. Our approach responds to data, but we use our expertise to look for issues from minimal studies. We often recommend rechecking solubility of new batches of API when solubility has been selected as a critical factor, especially if there's been a process or site change, as an example. We understand many small companies are increasingly under pressure to skip or minimize activities to reduce cost in order to quickly get to phase 1 proof of concept and to unlock more funding. As well as reducing cost, any activity that reduces timeline also helps reduce cash burn. This chart shows the traditional approach of developing an API, then developing a drug product afterwards. Some small companies look to skip the later stages of API development and plan to manage all the liabilities with a formulation approach, such as spray dry dispersion or lipid formulation, both which dissolve the crystalline API and make crystal form less relevant. Of course, this can be done, but if we skip the step completely, there are risks that we will not be able to develop the API at all, or maybe we will still be developing API during phase 2, which can play havoc with future clinical supplies. Within Catalent, we look to integrate the API development and selection decision-making process in order not to make these important decisions in isolation without the formulator, as this is where a lot of consequences are created. We raise potential issues and track them through the project in order to mitigate the consequences in an integrated way. The major issue for most people is solubility limitation, exacerbated by the need for dose escalation. New molecule challenges make powder or API and bottle first approaches theoretically unviable 70 to 90% of the time, but we still see teams ignoring or gambling on this point, even when there is little data upon which to base a decision. Inability to achieve the end result in dose escalation is a common reason for phase 1 failure. Advanced formulation expertise and technologies are required to reduce risk early on and achieve a viable formulation to increase success rate during dose escalation, toxin phase 1, even if it isn't needed for phase 2. The fastest and most efficient way to achieve a viable formulation is to conduct feasibility studies on relevant formulation options in parallel and select the optimized approach based on animal PK data. It may be possible to use a simpler formulation for phase 2, but you won't get there without collecting tox data. This slide covers the dosage form development integration, where we utilize a 5-step process to find the right molecule, select the right salt or polymorphic form, and once we have that know your molecule information, we move into technology section to the selection, the 3rd step, which is typically a parallel screen for a new chemical entity, then on into GLP tox supplies and to phase 1, first in human. The path of any one molecule through these steps will be unique, but working on many molecules, we see trends that help small companies avoid pitfalls. As well as physicochemical evaluation, we also look at PK absorption using expert PBPK modeling, which has generated huge interest since we incorporated this capability about 5 years ago. As problems with solubility, first pass loss, efflux, gut metabolism, can substantially complicate PK evaluation and are usually impossible to separate without the help of PBPK expertise. PBPK modeling can be applied at any time through the whole process, with the modeling usually getting more sophisticated and better as more data becomes available and more useful as we migrate through species into human. As I mentioned previously, we work hard to collaborate across teams to have a consistent approach around the globe. In early development, we typically work at sites that are designed to respond more quickly, such as San Diego and Nottingham. In early development, managing the evolution of the dosage form whilst meeting project technical, quality and timeline requirements is surprisingly challenging and requires expertise and communication. In late-stage discovery, we are focused on solubility and may use aggressive solvents. In GLP tox, we shift focus to exposure, but also need chemical, physical stability, ease of preparation and dosing, and 28 days data for safety. In phase 1, the focus is on safety, bioavailability enhancement, usually, GMP compliance, CMC support, scalability, packaging, labeling, etc. We engage quality assurance and manufacturing experts so that they can become familiar with the plan and provide valuable comments on any foreseen difficulties from their experience in how the interaction between Catalent and the CRO will work and to set up the right quality agreements. But the integration goes beyond the scientific approach into early development and extends into our one partner approach to downstream clinical manufacture, packaging, and clinical supply, as Mark recommended. This slide shows a typical linear timeline in drug development, beginning with development and manufacturing of early phase products, starting with candidate selection, where we identify the best candidate to move forward with, through relevant steps to dose form development and selection. As you can see on this slide, these activities are commonly performed in a siloed fashion, all the way through resupply of the treatments when needed. When we go through these phases step by step, as shown here and as shown by Mark, the time to get to clinic can take many months. Here we have the same activities as on the previous slide, but now integrated, especially initiating clinical supply activities with those of manufacturing, getting a major head start by initiating packaging design while still in the process development stage. The benefits, as you can see, are that the teams are working closer together, integrating their activities and supporting strong communication along the way. Having earlier access to critical information allows long lead term activities to begin earlier, so a sponsor isn't waiting for these to complete at the end. This approach also creates buffer to the schedule, allowing teams to effectively plan, mitigate risks, and avoid delays that may arise otherwise. This is important if the customer does not have their own expertise in the same technologies in order to accurately predict what the long lead time items might actually be and take some of the burden of communication away. Within Catalent, various teams all work together under one quality system, one confidentiality agreement, and work together frequently, reducing handoffs and points of contact across multiple vendors. Teams can anticipate future needs and improve communication across the entire project and shrink transition periods to save time. We have more than 100 active molecules using this approach from Catalent, showing the value of that our clients are seeing with an integrated one-team approach. We have a specialized team of 9 integrated project managers across the business and more than 50 project managers trained to support integrated projects, all of which means potential time savings. And all of this integration has been implemented in our latest offering for adaptive phase 1 clinical trials that is called Express Pharmaceutics. Adaptive trials leverage regulatory flexibility in countries like the UK to complete clinical assessment under a flexible quality by design, design space approach, rather than a rigid IND, IMPD supported drug product application. Using this approach can save considerable time and money, while at the same time providing a more robust end result. Catalent manufactures immediate drug product, which is filled into final drug product at the CRO's pharmacy for cohort dosing. A lot of people are interested in saving the time and money without the quality by design approach, but the reason they should be interested is to integrate formulation and clinical supply to deliver better outcomes as well. There is more than usual complexity in establishing exactly what is being done and who has the responsibility for what. Catalent's quality and regulatory affairs teams can help support customers who are interested in exploiting this offering. And Catalent has a partnership with Hammersmith Medicine's Research in the UK, who are an early pioneer in this field to ensure integration. There is a separate webinar on this topic that should be available if you'd like to know more. I've pulled examples from recent projects to highlight some of the typical issues we have seen over the last 12 months. In candidate selection, several customers have presented data like this one, where the choice of lead candidate was not so obvious, where there wasn't a clear winner. In a few cases, the final decision seems to have been made based on API manufacturability without any consideration for future drug product manufacturability or PK liabilities, and without the data to know whether these issues could be mitigated. In this case, we recommended hanging on to the backup molecule for longer in the project profile, although that would cost more money. In pre-formulation, many customers see API salt screening as the step before technology selection rather than another technology to improve solubility of an API. Several customers have asked for enabling technology formulation of a salt, which has at least two issues. One, it increases the molecular weight that needs to be solubilized and reduces solubility in non-polar solvents. And two, some spray drying equipment can be affected by strong acid salts like hydrochloride. Many teams want to skip pre-formulation completely, which is why I prefer the term material characterization, as we end up doing it later to understand cause of failure. In technology selection, we see many teams struggle with the problem of poor solubility. Here, we tested prototype formulations made by another team that performed poorly in a PK study. By testing both equilibrium and kinetic solubility, we were able to show that the problem was a combination of fast crystallizing API, difficult pH solubility profile, and failure of precipitation inhibitors to provide parachute. We remade prototypes that demonstrated improved performance using small-scale biphasic dissolution studies before progressing into animal PK studies. We routinely utilize small-scale dissolution apparatus with built-in UV probes using simulated media in a biphasic media switch protocol to be more relevant to the in vivo case. In GLP studies, and more broadly in animal PK studies, we help teams understand reasons for poor bioavailability and help with improving data quality and beginning to ask questions about what might happen in humans. In this case, the pH solubility profile of the molecule was rapidly changing at around the same pH that is found in the dog's stomachs, even after pentagastrin treatment. We suggested using a different animal after proving that it was difficult to manage in any formulation approach. In phase 1, we saw several cases where ascending dose study outcomes were disappointing and the cause was complex, with a combination of nonlinear efflux and gut metabolism and precipitation at higher doses. In this case, we were able to confirm the causes with a combination of PBPK modeling and material characterization, and then provided new formulation options with a parallel technology screen. As well as getting the project back on track, we recommended lipid solutions as the fastest path, not only to restart clinical studies, but also to market due to the more extensive development that would have been needed for spray dry dispersions or hot melt extrusions or other technologies. At the start, I said I would show you in high level how working with Catalent gives your molecule the best chance at success early. Integrating development disciplines provides efficiencies that are often overlooked. 1, improve communication. Focus on the critical urgent issues, thinking about the finish line and not just the start, sharing insights and recommendations from our expert networks in order to make sure everyone is aware of the plan and the issues to be addressed to stay on track. 2, improve outcomes. By applying best scientific practice and technology expertise earlier in the pipeline to flush out obstacles to be addressed that smaller, less experienced teams miss. Also, getting into the right technology from the beginning, thus avoiding need to change or repeat studies that lead to delay. And 3, shorten timelines. But balancing all factors and not prioritizing time over everything else, avoiding delay without simply rushing to start. I hope I've shown the benefit of working with experts and made a case for working with Catalent in order to boost the expertise, especially in small teams. I see too many teams with over-optimistic timelines that do not recognize or respond well to data showing that the planned approach is not likely to work. I'm convinced that for most molecules, a science-driven approach will eventually prevail. If you have a problem molecule right now, or if you're interested in hearing more about how Catalent helps many, many small customers, please get in touch. And with that, I'd like to thank you all for your attention.
All right, thank you to our panel for that. Let's move directly to our questions. And the first one, I'm going to read it verbatim here for our panel. When integrating multiple disciplines, supporting the advancement of a drug through early development and beyond, are there best practices that you can touch on, especially when the various work streams are operating out of multiple organizations? Mark and Stephen, who would like to take that first?
Steve, I can take that one. So yes, there are some good practices or best practices that can be used. As I alluded to, collaboration, coordination, and acting on information as it comes in is very important. And typically there would be a cross-functional CMC team that is in place to manage these activities and manage the information as it's coming through. In early development, it's a very steep learning curve. New information is coming through quickly. And even when all of those functions sit within the same organization, it's typical to use a cross-functional CMC team, meeting weekly, bi-weekly, depending on how quickly things are moving along, but to act on the information collectively. So if there's changes in API development, we're seeing a new polymorph, it can be discussed, which path do we take? Do we pursue a new polymorph? Do we do additional screens? How is this impacting formulation? It's really important to do that. These are typically led by people that have broad and deep understanding of CMC development, especially in the early space. And then you have the functional experts from those functions that sit on the team. That same team should also be connected to the overall drug development team that relates to clinical and the likes, because there are things that can come out of early CMC development, improved bioavailability, as I said, dealing with food effects and the like, that should be communicated back to the clinical group. Is that a benefit to us? Is that not a benefit to us? Are we seeing a very fast half-life? Do we need to stretch that out with delayed formulations or extended release formulations? So I think the answer is yes, having a core group of individuals from those various functional areas, even if it's in the company or even if it's spread across the world, meeting on a fairly frequent basis to act on the information as it comes in.
All right. Stephen, anything to add there?
No, I'd like to get to the other questions.
All right. Okay, let's get to the other questions then. Let's ask one of these over here right to you, Stephen, and given what you can reveal. So how often does Catalent use spray-dry dispersion in early clinical trials? And are you able to comment on the viability of this technology in commercialization?
Oh, thank you for the question. Yeah, so spray drying is the darling of early development for a lot of people. As I mentioned in my presentation, if you solubilize the drug in solvents and spray dry them, then you're avoiding all the issues that you might have with crystalline material, and you're making it amorphous. If you can stabilize that, you get great solubility as long as you can hang on to that. So this is a great technology for fixing solubility issues in early development. However, whether that transitions to a commercial product, there are difficulties in converting spray-dried material into a finished dosage form. And the spray process itself can be rather slow and uses a lot of solvents. So a lot of people should consider transitioning to hot melt extrusion to make the process a little cheaper. And if they're able to establish a dose that doesn't need spray drying, you might be into a simpler technology. So, definitely, I would say it's definitely a viable commercial technology. It's been shown there are a bunch of commercial products out there. It must be one of the most expensive ways of making a drug product. So if there's another way of getting the same result more cheaply, you might consider that.
Okay.
And Steve, if I can add on to that, just to your point, yes, there are several, quite a few, in fact, commercial products that use spray dry technology to make their formulation. It can be a bit scary or intimidating. You have an amorphous material, you have to worry about crystallization, but it can have tremendous benefits in terms of improving bioavailability for very poorly soluble molecules. However, it goes back to what I said. It's really understanding the physical chemical properties of the molecule, understanding how to control it, knowing if and when it's going to crystallize and how to make sure it doesn't crystallize and it stays amorphous as a spray dried dispersion. So it is commercial technology. A lot of companies are a bit concerned about using it, but with good understanding, it can be used.
Okay, so a question from the audience here. So when in the process do you recommend polymorph and salt screening? Before initiating API synthesis for tox or in parallel with the synthesis?
My answer is to do it early and do it quickly. At least start to look at the landscape, right? Because one, you want to be able to have as highly soluble molecule as you can. You also want to understand what are the more stable polymorphs, the more stable crystal forms. Every company has a story. Every big pharma company has a story about a new polymorph showing up very late in development, or even after a commercial product has been sold for several years. It's the case. So my advice is to get that done fairly quickly. Do a quick screen. Make sure you really understand what different polymorphs or crystal forms you have out there, and use it to your benefit if you need to.
Okay.
Yeah, I would add to that. Like, would you jump into a swimming pool without checking the temperature first? If it's freezing cold, you might regret that. Whether you have the money to do the extensive studies that have been done in the past is a question. So we do see a lot of people doing either no screening, that's very rare, or limited screening, as Mark has said, to at least collect some information, and then working with your solid state guys to work out whether this API is better or worse than other ones they've seen, and then using the best scientific practice approach that I've talked about. If you're dissolving the drug, making formulations in different solvents, different lipid excipients, you should always be looking out for that new polymorph. If I get crystallization in any of my systems, just double check those to look for new polymorphs, because that's a good example. And I didn't plant the seed question. It's a good example about how orthogonal data if you keep an eye on it, working with an integrated expert team, it can inform you about things that you weren't actually planning. And that's part of best scientific practice.
Okay, wonderful question. I'm now going to combine two audience questions here together for you guys. And I think this is really more for Stephen, but let me read the first one and I'm gonna read the second one and then you can react to this. So the first question is, could you further elaborate on the relative ratios on different formulations Catalent sees going through the phase 1 and phase 2A trials? And then a piggyback question to that was, in the presentation you mentioned you recommended lipid formulation for FIH. With what you can reveal, can you provide a little bit on the formulations? Is it a solid dosage form? What are the excipients? What are the MDI of the excipient, batch size, and so on? I think there's a lot of formulation questions here.
The first question, I don't understand. Ratio of what? Ratio of what is that? So if the guy who asked the question could tell me, the ratio going into phase 1 and phase 2A trials. Well, there's a lot more going into phase 1, if that's the ratio you're asking about. There is I don't have the data to my fingertips. Mark, do you know?
No, I don't.
There was a tough study a few years ago that had that kind of information. I could dig that out. I'll commit to sending that after the presentation.
Okay. All right.
That's not really Catalent data, though. I don't know that I have that data in my fingertips. It would take me a bit of time to get that information.
Let me ask you a Catalent specific question here while we come up with that right from the audience here. What is Catalent and NGT's capability for supporting development of different modality assets such as ADCs, vaccines, nanoparticle delivery systems, and cell-based therapies?
I mean, quickly from an NGT perspective, yes, we can supply that expertise to do it. As I suggested, it's consultants who understand both that space with years and years of experience in terms of depth, and then that cross-functional connectivity in that area to be able to make sure you are taking that integrated approach and address some of the issues that we've talked about this morning.
Yeah,
Right.
from a Catalan point of view, it's easier to say what we don't do than what we do, because I think everything on that list we can do. The one technology that we are not commercial in is nanoparticles for small molecules. So we can do it in like a preclinical setting. When we look at the utilization of nanoparticles over the last few decades, there are not that many products that have had a side-by-side comparison done to show the relative benefit of nanoparticles over other technologies. So we think that most people prefer spray drying over nanoparticles because it's about the same complexity with a better result.
Yep.
So we do have nanoparticle capability in our organization in preclinical settings. So we know a bit about how difficult that is.
Okay. A question from the audience here. I have been using HME technology for solubility as well as development of personalized medications, but the excipients process is a big problem. How to solve the processing problems?
There are only a handful of excipients that are typically used as the main polymer for HME technology. If you're using them, you shouldn't have any problem. If you're adding excipients to an HME formulation, to add plasticization, if you're adding other excipients to the HME melt itself, that may be where you can get into problems. I can't really. Other than that, HME is a great technology for making drugs into the amorphous phase. The big advantage over HME, over spray dry dispersions, is that the particles that you produced, if you mill them to a relatively larger particle size, the ability to convert them into a blend for capsule filling or tableting is better. So HME is normally easier to convert into a finished dosage form. So if the person who wrote this question wants to know a little bit more, I can connect them with our HME expertise team based in Somerset, New Jersey.
Wonderful. Yeah, and all attendees will follow up with an on-demand version of this and get in touch. And from Endpoints, we can get you in touch with Catalent as well. No worries about that. Here's a question, rather broad one, I think, but let's get your guys' take on it in the context of today's webinar. Could you elaborate further on the pros versus cons of adaptive versus traditional clinical trial strategies?
That was your slide, Mark.
Well, I'm not sure it was, but that's okay. Adaptive trials
question. Right.
Yeah, I mean, adaptive trials have the ability of being able to adjust and basically act in real time, depending on what you're seeing in the clinical sense. So they're being used more and more, but it's also molecule specific. So I think it depends on the business case and the molecule for a company.
So yeah, sorry. When I saw the word adaptive, I read integrated. Sorry, Mark, that was my bad. So in a traditional clinical study, you create a formulation based on what you've seen in the GLP in the preclinical setting, and you get permission to study that formulation. You have to collect stability data for that, and then you put it into a dose escalation where you must follow the protocol that you have approved with the regulators and with the ethics committee. So you're fairly straitjacketed in the traditional approach, which most people understand pretty well. But suppose the humans are not well represented by your animal model and there are some surprises, or maybe you had some data in the preclinical setting that made you think that maybe the molecule would be a substrate for efflux and maybe it would not, and you didn't know which way to go. In the traditional way, all you can do is throw everything like a kitchen sink formulation, everything into the formulation and evaluate that. And you will not know at the end of the study you won't be able to tease out any information about the role of different excipients in the formulation. In an adaptive trial, you can respond to the data from the clinical trial, and maybe first thing you can do, you give a dose range, not the actual escalation, and you don't have to. You have to start with the lowest dose, and you cannot exceed the highest dose. But you're free to change the intermediate doses based on what you're seeing. In addition, you are also able to, in theory, change the formulation depending on the regulatory agency you're working with in order to respond to the issue I said. So if you see you were guessing that efflux might be a problem, and you can start the study without the efflux inhibitor, and then halfway through you could incorporate one because you're filling blends at the CRO, right? And then overcome that efflux inhibition with a formulation change in the middle of the study, providing that protocol was approved by the regulatory agency and by the ethics committee. It should be possible to do that.
All right. Let me ask you guys, so how accurate is PBPK modeling? And in your experience, why should we be trusting a computer algorithm for
that? So it's called PBPK modeling because the computer's creating an algorithm to match what goes on in people. So if you only have one data set and you get a match, you can kind of score whether your R-squared value, you'd call it on an Excel graph, but you can kind of judge how closely you match that protocol. Whether it's robust or not will depend on the quality of the data. That can be a bit problematic in early development. Secondly, it's probably better to plug in data from several studies from several species to challenge the model and make sure, because the interplay between solubility and permeability can play out in a computer model and look the same. So like the more you challenge the model, the better, which is so from an in silico, just taking a molecule and doing a PK modeling, that's kind of, it can be more or less accurate depending on how close that molecule is to the training data set that was used for the molecule structures in the data set. And then how good it is, how good that molecule is, depends as much on the expertise of the person doing it as on the model themselves. Okay. And so that's where challenging the model and making sure that, you know, in the same way as a critical process parameter, right? If you have a set of rails, like a railway line, like I showed you, and you never deviate from those rails, you don't know your points of failure. So one thing we would encourage you to do when any study is to wobble a bit and see if you're still safe. That's what I would recommend for PBPK modeling as well.
Wonderful. Okay. We have just time for one last question. And my question, I think it's mostly for you here, Stephen. You work at a company. We've done multiple forums like this together. You work at Catalent. Catalent's got a lot of capabilities, a lot of expertise, a lot of footprint. So the question here comes from a lot of smaller companies here. Is Catalent really work with smaller companies? And are there benefits to that for a smaller company to work with Catalent?
Well, the answer is yes. I work with small companies all the time. Surprising how many small companies we work with. Our feeling is that small company molecules will one day grow up and be big company molecules, and we like to be involved early on so that they have a better chance of becoming larger projects in the future. So, like, you know, we want to help people across the whole gamut of sizes. I think that's the best answer I can give. I think part of the question, the second part of the question, Arif, did I mention that as well? Did I cover that?
You did. I mean, are there benefits to working with smaller clients, I suppose?
Yeah. So it was in my slide deck, actually. Yeah, that's what I'd forgotten. That's right, in my slide deck. If you have a team of 2 or 3 people, you can tap into a company with like 2,000 scientists, you know, and ask their opinion. And that's part of the scientific advisory. If you have a question, you're interested in evaluating that with Catalent, you can talk to the scientific advisory and get an opinion anytime.
All right. Well, that's all the time we have for today. And I want to thank everyone in the audience foremost for tuning in. And thank you both, Stephen Tyndall and Mark Crook, for sharing your time and expertise with us today. And thanks again to Catalent for sponsoring the discussion at Endpoints. If you want to rewatch this webinar or share it with your colleagues, a link for on-demand viewing will be shared tomorrow. I'm Arslan Arif for Endpoints News. Thanks for joining us, and we hope to see you at a future event. Thanks, everybody.